They're selected for gram-negative cultures because they contain chemicals that inhibit the growth of gram-positive. So they're both selected for gram-negative. If you had growth on T7 or MacConkey's, that should have been gram-negative. We had a couple of plates that I looked at last time that were kind of suspicious. It looked like some of our gram-positives may have snuck onto those plates. They're not supposed to be able to do that. Apparently nobody told them that. But because these are selected for gram-negative, what you have on those two media types should be gram-negative. And then, the other thing that we know about these is that they're differential. So what's the basis for differentiation for MacConkey's and T7? They contain lactose. Lactose is the carbohydrate present, it's a disaccharide, and if the organisms can ferment the lactose, typically they will produce colonies that are brightly colored. Or you can think warm colors, pink or yellow, as opposed to blue or kind of tan. The pH indicators vary. So Churchital 7, the pH indicator is bromothiamyl blue. For MacConkey's agar, the pH indicator is neutral red. It says in there. There we go. Bromothiamyl blue, neutral red. It was important that you record the color, not only the color of the colonies, but the color of the medium behind the colonies, because it tells you something. It tells you, can they ferment lactose or not? So hopefully you recorded that. Mueller-Hinton agar and Nutrien agar, they're not selective. They can be differential. Some of you have cultures that produce a water-soluble pigment that diffuses into the medium. Mueller-Hinton agar doesn't start out green, and there's quite a number of people that have these nice green plates now. Can I hold that up? Greenness. That greenness is being provided by the culture itself, because Mueller-Hinton agar doesn't look like that. T7. Sorry. Well, it does look a lot like one. I see a lot of them though. That's T7 medium. That one back there. Did you hold yours up? There you go. Right. That greenness is not in the medium to start with. This one was differential. Anyway, so you had to look at them kind of closely. Blood agar's not selective, but it is differential on the basis of hemolysis reactions. Several of you have cultures on blood agar, and so we need to look at the hemolysis reactions associated with blood agar. I don't think I have pictures of them up here. Mannitol salt is selective. What is it selective for? Aliphiles, because it's salt. It has 5% salt in it. It's 7.5%. 7.5% sodium chloride. The cultures that we are trying to grow on here mostly are staph. So Staphylococcus is a halophile, and here we see Staphylococcus aureus and epidermidis growing on there. It is also differential. What's the pH indicator? Phenol red, and it's differential on the basis of mannitol fermentation. It's handy, it's in the name, mannitol salt agar. It has mannitol in it. If they ferment mannitol, they produce acid and turn the phenol red indicator yellow. If they don't ferment, that doesn't happen. So what you were supposed to do, this is where we were last time, and we ran out of time. You were supposed to treat 2 new plates and establish pure cultures of the organisms that now you're going to go forward and identify. You have to identify both of them. So theoretically, that has happened. Hypothetically. Anyway. If you turn to page 167, there's a guide to the identification of unknowns. If you look at this on the website, it's in color. Where it's in the lab manual, it's all black and white. So we're going to use this information and the information gained from your gram stains. How many of you got to look at your gram stains? Most of you did. Okay, good. Some people, I think it was just too crazy. So, first of all, these organisms were divided into 2 general categories based on gram stain. This identification guide is kind of a dichotomous key where you answer one question. The first question is, what is the gram stain reaction? Is it gram positive or is it gram negative? And then you go from there. So group 1 are gram positive and they're COXY. In the group 1 set, You can have organisms belonging to the genera Staphylococcus, Streptococcus, or Enterococcus. So we have 3 possible genera here that are gram-positive cocci.
Oh, can you go back a little bit to the staph, strep, and what?
Go back. Wait.
Oh, 167.
Yeah. So, group 1, gram-positive cocci. The possibilities are Staphylococcus, Streptococcus, Enterococcus. That's on there, but actually no. I could have put those out, but I don't. The micrococcus organisms are usually brightly colored and they're just too easy to identify. It's like, oh, those are red ones, Angeles, those are yellow ones, that's Micrococcus luteus. Actually, Micrococcus angeles is now Arthobacter anyway. Okay. So, if you have gram-positive cocci, the first enzymatic test that you want to apply is called the catalase test. The catalase is extremely easy to do. What you do is put a little gob, a visible gob of your cellular material on the surface of a clean glass slide. If you work with somebody else, you can put more than 1 sample on the same slide. And then you put on top of it 3% hydrogen peroxide. This becomes a place in lab where you really have to pay attention to labels on bottles. Because the 3% hydrogen peroxide is always hiding in the refrigerator. And there's another bottle that's often out in the green trays, and it's 3% potassium hydroxide, 3% KOH. If you try to do a catalase test with 3% KOH, it will not work. It'll give you a false negative every time. The catalase test, think about catalase enzymes and what they do. They break down hydrogen peroxide, H2O2. When you split that, what happens is oxygen comes off as a gas and water is left over. So you get a very strong bubbling reaction. You just put 1 little drop, hydrogen peroxide, on the surface of the cells. If you get a lot of bubble formation, that's catalase positive. So if you have gram-positive cocci, you should run a catalase test. Be careful if you're taking your samples off a blood agar, because the blood cells that are in the agar will give you a false positive on the catalase test. So you have to be really careful that you're only picking up cells. So our catalase positive cultures should be growing on mannitol salt agar, whereas our catalase negative cultures are growing on blood agar. That's rather a big hint. So you can think about that in terms of what medium type you inoculated to begin with. So mannitol salt agar is on the left here. We did this for our replica plating exercise. Staphylococcus aureus is at the top, Staphylococcus aureus is at the bottom. One type ferments mannitol, the other does not. On blood agar, we have either Streptococcus or Enterococcus, and they are the little white colonies, as opposed to the bigger kind of gray or tan colonies. But the bigger tan or gray colonies, if you take something off a blood agar and it gives you a catalase positive, it's the wrong culture. It's not what you're supposed to be working on. Now, if you have organisms that were growing on mannitol salt agar, you have to do the catalase test. You also have to do a test called the methyl red Voges-Proskauer test. I'm going to set up a series of media types, and they will be up here on the front bench because I don't know where else to put them. The methyl red Voges-Proskauer medium is liquid, and you inoculate 1 tube. To inoculate it, you just pick up a visible blob of cells and put them in there, shake them off your loop so that they're in that medium. This ends up being 2 tests. One is testing for acid production, that's the methyl red test. The other is testing for the production of acetylmethylcarbinol, or acetylene. We'll talk about how you evaluate those tests next time. But if you have gram-positive cocci and they were initially growing on mannitol salt agar, last time you were supposed to restreak them onto TSA. Use the sample from TSA to run your catalase test and to inoculate the MRVP medium. And I'll put all this on the board. The other thing you're supposed to know is, do they ferment mannitol? How do you find that out? Yeah, you just look at your MSA plate. sometimes people wanna inoculate another tube of mannitol. It's like, no, don't go there. You've already got mannitol. It's on the MSA plate. Just look at that plate. Is it yellow or is it not? There is one other test you need to run, and it's called the coagulase test. Now, coagulase is another enzyme. Notice the ending. So ASE, it tells you it is an enzyme, and the coagulase enzyme coagulates something. Turns out what it coagulates is blood plasma, and what we have for blood plasma is rabbit. You can't squeeze a rabbit. Comes in a bottle. So our coagulase plasma that's interesting. It says coagulase good, and then it has dates. One is 10/3/17 and the other one is 10/18/17. Well, I guess some of these were made up fresh and some of them weren't, and you have no way of knowing which ones are which. The coagulase plasma, it's just rabbit plasma in a tube. It's liquid. To inoculate this, again, you're going to put a visible amount of cellular material in here, and then we incubate it for 24 hours. If the organisms can produce coagulase enzymes, this liquid will become solid. It's blood plasma, and they will coagulate it and make it solid. So, how many people know they need a coagulase tube? If you had organisms on mannitol salt fogger, you need a coagulase tube.
Anyone? Would it be possible that you had gram-positive toxin that didn't put on mannitol salt offer?
Get your own blood over. No?
TSA and MH, wait, MHA? No, you have TSA. Try all your hair off your feet there.
According to my grandma's game.
She was right.
Anybody else? If you have gram-positive toxin on mannitol salt dogger, you need a coagulase tube.
Do you want the spoonful from our new leaf or the one from the?
You inoculate it from your TSA plate, because those are the freshest stuff.
Plasma or something.
Well, didn't you do it right there? That's right, but you really need to look at them because you have a possibility that the other organisms might work.
I want to drink.
So everyone could just talk to
Trying to get you to look at them.
No, I know, I was gonna look at it today.
Okay, what I'm wondering is if I accidentally
No, don't go there. Assume you're right. Okay.
Don't doubt yourself.
No, don't doubt yourself. Okay, so everybody got that. We got the coagulase out for the people that need it. So, again, I'll put this on the board. Now, if you have cultures that are growing on blood agar, again, you need to do catalase test, assuming you have gram-positive toxin on blood agar. You do a catalase test, and then you're going to inoculate esculin medium. We have two different sets of esculin medium. One has bile salts in it and one doesn't. We're going to use the esculin medium that does not contain bile salts. The esculin medium is in a tube with a slant surface. I probably am going to have to go round these up because I don't know where they are. So I'll just tell you, a slant surface is going to be at an angle like this. To inoculate the esculin, you put a sample of your organism on your loop, go to the bottom of the slant, and do a fishtail streak up the surface of the slant, and then also stab to the bottom. So we're just trying to get our organism onto the esculin medium. Again, this is if you have gram-positive cocci on blood agar. And then the other thing that you need to look at is, can your organisms hemolyze red blood cells? Well, this is described in the lab manual, but let me just go over it real quick. Hemolysis reactions, initially carried out to distinguish the different types of streptococcus. The problem that we run into, there are different types of blood agar. So what we use in this laboratory is sheep blood, and some labs use horse blood, some labs use bovine blood. So you can get different reactions from these. But the reactions of hemolysis are called alpha. Indicated by the Greek letter alpha. If organisms are doing a really nice alpha hemolysis, the agar behind the colonies turns green. I'm going to say greenish. Because our alpha hemolysis reaction is not real good. It's kind of greenish, it's kind of yellowish, but it's definitely, it's going to change the color of the medium. If the organisms show beta hemolysis, Greek letter beta looks like this. This is complete lysis of red blood cells. If the organisms are doing alpha hemolysis, they're simply going to convert the hemoglobin to methemoglobin, which is green. But if they're doing beta hemolysis, they're going to lyse those red blood cells completely, and the medium will be clear behind the colonies. This can take a while. It should be completely clear, but it may take several days to do it. The other hemolysis reaction is gamma. And gamma hemolysis is no reaction. No change in color, except that all the blood agar, when you put it in the incubator, tends to get a little more brown than red. It's very bright red when you put it in. When you take it out, it'll be a little bit brownish, but there's no change in color behind the colonies relative to anything else on the plate. So if you have colonies growing on blood agar, you want to look at them and see what your hemolysis reaction is going to be. Okay, moving right along. The next set, group 2 organisms, gram-positive bacilli. Sometimes these organisms are a little bit difficult to isolate, but if you see them in the gram stain, you can get pretty close to identify them just looking at their morphology. First question is, what are the gas requirements possible for cultures that are grown in the atmosphere? This was on our last quiz. What can we not grow, either in the drawer or in the incubator? Obligate anaerobes. We can't. There's oxygen in the air, there's oxygen in the incubator, in the drawer. So we can't grow obligate anaerobes, which means the organisms we're growing are either facultative or they're obligately aerobic. They typically show up on the nutrient agar looking somewhat different than colonies that are circular and convex. Sometimes they're spready and textured on the surface. Anyway, they are growing on nutrient agar, and I told you to keep them on nutrient agar last time. If you are identifying these guys, you need to know, can they form endospores? You do not need to do an endospore stain. You can see endospores just fine with a gram stain if the culture is producing them. And that's where we run into trouble, because sometimes they do and sometimes they don't. If you're looking at really fresh cultures and you don't see any endospores, then try looking at an older culture. See, did they get around to making them? Eventually they will. If you find endospores, you need to know what's the shape. Endospore shape is either spherical, which is round like a ball, or ellipsoidal, which is more jelly bean shaped. You need to know the location. Is the endospore central or terminal? And you need to know, is the sporangium swollen or not? Now you can determine this even if you don't get these guys isolated. Just look at your gram stain. Then you're supposed to measure them, get an exact cell diameter, and you want to know, is the arrangement streptobacilli or not? If you're measuring, what's the most accurate stain to use? Indirect. Indirect. So last time I had you guys do gram stains, you do need to do indirects for your 2 physiological unknown organisms also, so you can measure them. So the identification then is based on cell size, endospore location and shape, arrangement of the cells, and you really don't have to do any enzymatic testing for those guys. The next group are the groups 3, 4, the gram negatives. The gram negatives are put together primarily because it's really difficult for students to distinguish cocci from bacilli, because our bacteria are pretty small, and short bacilli can look like cocci. Also, if they really were cocci, when they're getting ready to undergo fission, they elongate, then they look like bacilli. So we just put them all together. So, group 3, 4. The first test that you need to do, and this is why I pass them out to certain people, is the test that tells us what kind of metabolism are these cultures running. Are they fermentative, or do they have an oxidative metabolism? So this is the oxidation fermentation test, or OF test. So this is used to determine the type of metabolism. If you have a semester project going, and your semester project organisms are gram negative, you should run an OF, because this is a test that you need to run to find out what kind of metabolism they have. Beyond that, some of you are going to have OF tests to look at right now, and some of you will have to set this test up tonight. This medium contains the pH indicator bromophthalmol blue, so it's green to start. Green is the neutral color with that pH indicator, and it contains glucose. Glucose is the carbohydrate present. There are 2 tubes that are inoculated for each organism type. One tube has an upside down glass tube inside the medium, it's called Durham tube, and one doesn't. The easiest way to tell if there's a tube in there is the cap color. Because the people that are prepping our media now have been really nice to us. If the OF media have green caps, they have tubes. If they have white caps, they don't. And they're labeled on the baskets as well. Let me mix those up for you. No. So, to inoculate the OF medium, you take a visible sample of organisms on your loop. And this is important too. Notice I'm telling you every time you inoculate these media, use a loop. Don't use your needle. Some of you have the straight wire. Don't use that. Use your loop because you want to get a good amount of cells. And you're going to put them to the bottom. We want to know if they can ferment. And then the tube that doesn't have the Durham tube gets sealed with Vaspar. So this is a liquid that goes on the surface. Here you can see this layer of white. It looks like wax. Well, it is wax. It's paraffin mixed with Vaseline. The reason we put the Vaseline in it is because it'll melt at a much lower temperature than paraffin by itself. So it's less dangerous to work with. It's kind of messy. The good news is you don't have to do anything with it except look at it. I do this, by the way. One semester, I used to try to get students to do the application of Vaspar until a student dropped the Vaspar container off the counter onto the floor. It was like, okay, we're not doing that anymore. It was just way messy. The carbohydrate in the OF medium is glucose. So we've been talking about, in lecture, we're talking about catabolism of glucose. Do organisms ferment or do they use cellular respiration? Glucose, if organisms can ferment, they can probably ferment glucose. It's a really abundant carbohydrate. They're very likely to be able to use it. So if they ferment, they will produce acid. But remember what I said about these guys. They are heterofermentative. You don't know what kind of acid they produce. We just know they produce acid. That will cause the pH indicator to turn yellow. If they ferment, they can grow under the Vaspar seal. Fermentative organisms can run their metabolism without oxygen being present. They ferment. So if we look at the two tubes and we see that they are yellow throughout, these were inoculated last night. This is supposed to be a 48-hour test, which is why there's a little bit of green on the inside of the derm tube here. But you can see both of these tubes are yellow. They grew under that Vaspar seal. No oxygen could get in there. So we know they ferment. They produce acid. This is a fermentative culture. Culture A. Now, if they are not fermentative, they may be able to produce some aerobic acid. So what we do is we look at them and say, what's happening right on the surface? These guys are producing some acid. It's right on the surface of the tube. They can't grow under the Vaspar seal, so we know that they are not capable of fermentation. But if they're running a Krebs cycle, the Krebs cycle is all about producing a bunch of organic acids that can diffuse into the medium. So these guys are producing aerobic acid, but they are respiratory. So seeing on the screen, here's the Vaspar seal. Oh, one other thing that's being produced here. So this is acid, but notice this gap. When that Vaspar goes in, it's liquid. It's molten. So it goes in as a liquid. There's no way I can make that hang in the air. The organisms have produced gas and they're pushing the Vaspar up. Here, they're not doing anything because they can't grow under the Vaspar seal. These organisms are respiratory, they need oxygen, but they can produce acid over here on the surface. So those of you that have the little racks, you need to look and see which culture is fermentative and which is respiratory. You may see, this is showing you the pH, the changes in color of the pH indicator. So bromthiamyl blue is green when it's neutral. The organisms produce acid, they'll turn it yellow, but if they produce alkaline end products, they can turn it blue. And we do have some organisms that will produce alkaline end products in the OF medium because there's peptone in the medium. It contains glucose, but it also has peptone. So, I don't know if I have a picture of it. No, I don't. Does anybody have one that's blue on top? There we go. So here we see a respiratory organism. The tube that goes with this is green. They can't grow under the Vaspar. And on the top, they've turned the medium, they've turned the indicator bromthiamyl blue, which means they're producing an alkaline end product. So this is an alternative thing that they can do. I have a picture of it somewhere, but that works better anyway. Now, everybody has 1 gram-negative fermentative culture, but some of you guys have 2 gram-negatives. Guess who? The ones that I gave the baskets to, because we had to have this data in order for you to know which ones are fermenting and which ones are not. The rest of you have 1 gram-negative fermentative and 1 something else. Gram-positive on nutrient agar, gram-positive on blood agar, gram-positive on mannitol salt agar. Something that I gave you that I shouldn't have, it's completely wrong. We do have some things on brain heart agar, which, yeah, we have to think about those ones too, but, okay, so everybody had something fermentative. Initially, it should have been growing on McConkey's or T7. Hopefully everybody got something growing on there. And then what were you supposed to put them on? TSA or Mueller-Hinton. So they should have been transferred to something else. Now, we are going to be doing quite a number of enzymatic tests on these gram-negative fermented cultures. Tonight we're going to start with a few tests. One of them is OF. So you will have to inoculate OF, unless I already gave you those two. You're going to use the culture from the TSA or Mueller-Hinton agar from your fermentative culture. Don't take them off of the, don't take them from the T7 or McConkey's plates, because those plates were actually streaked with a mix. And even though one thing grew, the other thing is still there. So you want to make sure that you're working with a pure culture. So these are 48-hour tests. Again, the OF tests, if I gave them to you, you don't have to do it again. If I didn't give them to you, you do have to do them. The OF test requires that you inoculate 2 tubes one with a germ tube, one without. One green cap, one white cap. You inoculate by putting the culture to the bottom. When you're trying to inoculate the tube that has the germ tube in it, people want to know, do you go into the germ tube? The answer is no, it's upside down. If you try to go into it, you're going to hit that glass. Can't. You have to go down beside it. You do want to put the culture to the bottom. And then we're going to collect those in a basket. I'll put some baskets up here. Well, first of all, I'm going to have to put the baskets up here to have the media, and then toward the end of the lab, I'll set up another set of baskets to collect the media you have inoculated. So you have to do an OF. The next test you need to run is an MRVP. This is 1 tube of broth. Everybody should do 1. If you had a culture growing on mannitol salt agar, you have to do another 1. So people that have something that ferments that's gram-negative and something on mannitol salt, you're going to do 2 MRVP tubes. Don't mix them up. You need to keep track of which one's which. The next thing you're going to do is citrate. We're looking to see, can the organisms take citrate from the medium across their cell membrane. If they can, they have an enzyme called citrate permease. This is another slant where you streak the slant and stab. And I'll put this information on the board. The next medium we're inoculating is called urea agar. It's a slant. All you do is fishtail streak up the slant. And then we are inoculating the amino acid and amino acid control. The amino acid we're inoculating is lysine. The tube that is control just contains glucose. The tube with the lysine in it contains glucose and lysine. These 2 media types, the control and the amino acid medium, they look exactly the same. So it's very important when you pick these up that you put labels on these tubes. And these are broth, like the MRVP medium that's broth. You just pick up a visible blob of cells and put them in there. So we're going to inoculate tonight. Most of you are inoculating OF medium, that's 2 tubes, methyl red, 1 tube, unless you have a culture on mannitol salt, in which case you have to do 2. Citrate, urea, lysine, and the lysine control. Which culture are you using to inoculate those? Gram-negative, fermentative. If you don't know which one it is, please ask, because you do not want to inoculate all those media with the wrong culture. If you have non-fermentative gram-negatives, do not inoculate those media I just talked about, because it's a waste of time, and it's a waste of media. And the people that work in the back and make all these media get kind of hot under the collar when students waste media. Please don't. We have too many lab sections running. If you have a non-fermentative gram-negative, and some of you know that you do because you have the OF test to look at, what do you do with them? The oxidation fermentation test has been run. The next test you have to do is called an oxidase test. The oxidase test is run on a piece of filter paper that will be set in a glass petri dish. Filter paper is sitting in there. You're going to use a clean toothpick to pick up a sample of your culture, and you rub it into the reagent on the filter paper. It looks kind of like that. That's a picture of the filter paper with several spots that people have rubbed culture on there. If the culture is oxidase positive, and what we're testing for here is cytochrome C or cytochrome C oxidase. Anyway, if they produce the enzyme, the spot will turn purple. If they don't produce the enzyme, the spot should stay just kind of yellowish if you're taking it from TSA or Muir-Hinton agar. If you get this kind of a reaction, you probably took your sample off of a McConkey's plate and you shouldn't have. Okay, so that's the end of that presentation, but you know what? There are a couple of samples of organism types that I forgot to mention last time. Because we have gram-positive bacilli that are not spore formers. For the gram-positive bacilli that are not spore formers, you have to do a nitrate test. And we also have gram-negative fastidious that are on Reinhardt agar. And you don't do anything with them because they're very, well, you can do an oxidase test. Okay, I'm gonna erase this. You guys think about which culture you're going to use to inoculate all these media, okay? I'm gonna put the gram-negative bacilli up first.
I only get to do that. You can move something. What are we doing?
I didn't know the other
Yeah.
I didn't know the other
I
ones.
have gram-negative.
I have one.
You didn't separate them?
I
Exactly.
couldn't. Mine fucking died. And then she told me on Tuesday, she's like, I'll give you the.
Can I have your attention
See,
again?
she made
I
it
was
wrong
just
before.
asked a question, and the person came to the correct conclusion. Everybody has a gram-negative fermentative? Yes. So, if you have something growing on nutrient agar that's gram-positive, your gram-negative is gram-negative fermentative. If you have something growing on mannitol salt agar that is gram-positive, your gram-negative is fermentative. You have something positive on blood agar, your gram-negative is fermentative. Get the picture? Anyway, everybody has one, gram-negative fermentative. You're gonna inoculate the OF.
Sorry, I'm lost. I know it.
Is this sufficient to tell you what you're going to do? For the OF test, you get 2 tubes, one with a green top, one with a white top. You stab a sample of your culture to the bottom in each one. Some of you don't have to do that because I did already. So if you got one of these, that is your OF test, don't do it again. If you didn't get one of those, you have to do that test with your gram-negative fermented culture. Okay, citrate. You get a citrate slant. You streak the slant, fishtail, up the streak, up the slant, and stab it to the bottom. Urea. The urea auger, fishtail streak up the slant. Do not stab. So then you have to do a lysine and a control tube. They look exactly the same, so label them when you pick them up. You're going to add a blob of cells. These are bronch media. This is a highly technical term, a blob. It just means you have to see it. Make sure you can see what you're putting in there. you're gonna inoculate 2 tubes, just put a blob in. And then last one is Alphabet Boat Cross Tower. Now, if you had Staphylococcus, okay, if you had gram-positive cocci on mannitol salt dogger, but now they're on TSA, you need to do that for them also. Don't mix your cultures up. So you're gonna do 1 gram-negative fermented and 1 for your gram-positive. Everybody has to do this. So this is the most important thing. What I'm gonna do is put the media up here on the bench, the front bench, so you can find them. So that you can start doing this, and then I'll put the additional information up about other kinds of tests you need to run. These are the OF media, OF tubes. Notice the front of the basket. It says OF with tubes, OF without tubes. If you're running an OF test, you have to have one of each.
I don't know, I'm not even listening. No, I didn't mean to watch on time.
Well, I can't tell if she's something that's pure or something. Still better.